So what I'm going to talk about is I think we have to set the stage, no? And explain the problem, and then ask us the question, how can we afford this or not? And I was thinking to begin with this slide. And this slide, I like it because I think it sets the stage about what you were saying about cost effectiveness, right? So I'm going to talk a little bit more about glioblastoma because this is the tumor that I am working on. But I'm going to use glioblastoma as a model for other tumors that we have, that are aggressive tumors, and we don't have good therapeutics approaches against them. Look at this curve. I mean, this is the standard of care for glioblastoma, and this is what temozolomide is doing on top of radiotherapy. So here is what we have treating patients, temozolomide, a very expensive drug, and this temozolomide is just improving survival of patients for some months. So it's clear that we have to get to identify new therapeutic targets, new targets, or new therapeutic approaches against glioblastomas. And this is clear that we have to do it based on translational research, understanding the molecular mechanisms of the tumor to find better targets. But the situation right now is not very good. All right, this is what temozolomide is doing on most of chemo and radiotherapy agents, DNA damage agents. I mean, these agents, what they do is target proliferating cells, they do DNA damage. And this, we know it works, but it doesn't work too well, especially in aggressive tumors. So we all know, and I'm using this very old slide, that by understanding the molecular mechanisms implicated in tumors, involved in tumors, we will be able to find new therapeutic targets and then design new drugs. And this is something that we have to do, but it's also very is very costly. And then we need to do translational research. We know right now that the multidisciplinary effort of bringing together the basic science and the clinical science is very important in order to improve the treatment of the patient. And this has to be done at many different levels: genomics, transcriptomics, proteomics, studying the cell biology of these tumors, and also animal models, all of them in close relationship with the patient in order to improve the treatment of the patient, and also the research that is done in patients in clinical trials. It's very important that the information getting from those clinical trials can go back to the basic research. Also, it's a process that is very important, but it's also very costly. But this is something that we have been doing for a long time, and I'm using this slide in glioblastoma, an old one. And these are all the clinical trials that were being developing in 2007 in glioblastoma. The bad news is that all of these trials failed, and well, what's going on? And also going back to always the same question, I mean, how much money has been invested in these clinical trials? Well, we can discuss many reasons. One is the pharmacological characteristics of the compounds were not good. Maybe the therapeutic target was not a good one, or maybe there were some other problems in the design of the clinical trials. But now we are beginning to understand that one of the problems is intertumor heterogeneity. So tumors are different, and again, using the glioblastoma model, we know that each patient has a different combination of mutations, and also what we call glioblastoma can be subdivided in different molecular types. So that has a very important implication, and that means that we really need to characterize each tumor to know all the mutations that the tumor has, all the characteristics, the expression characteristics of this tumor, in order to find which is the better target and the better treatment. So we are talking about the tailored medicine. But something to take into account is that in order to get the best therapy, it's important to do a very, very thorough molecular diagnosis. So we need to do genomics, we need to do transcriptomics, we have to look at the proteins, and maybe also looking at other markers in the blood, like circulating markers. This, again, and going back to the question, is also very costly. And once we get this, we have to find which is the proper target, proper drug for this particular tumor, and then identify the patient. So we have success stories. I mean, we have seen in the previous talk by Michael Stratton some very good results about how to find new targets and, with compounds. And here we see some examples such as BRAF, BCR-ABL, and others. And we know that some of the drugs that are targeting these targets, these therapeutic targets, are working only for a period of time, and that's also very important. We know that there is acquired resistance. Even if the drug works, after a period of time, this drug tends to not working because of resistance. And in this case, for example, it can be due to the fact that tumor cells are alive and they progress in a way using the Darwinian evolution, and they can find mutations that can make them escape to the treatment. And a very nice example is the example of imatinib Gleevec, and how the kinase of ABL is acquiring mutations to escape to that. And that has also very important implication. That means that we must get a combination of treatments to target the same target in order to decrease the probability that the tumor cell will be able to escape to that treatment, again increasing the cost of the treatment. In the same way, now that we are using cocktail treatments for example, HIV and other infectious diseases. This is the example I was telling you, how the ABL kinase is able to mutate the cleft, the catalytic cleft, to inhibit to repress or inhabilitate the compound to get inhibiting the kinase. And then we have another problem. It has been described in a very recent paper that tumors, they are not only different between different patients, but within the same tumor, cells are different. At the levels of genomic, we have different mutations. That means that we will have to get serial biopsies in order to find out this heterogeneity, this intratumoral heterogeneity. So the molecular characterization of the tumor has to be also very, very thorough in order to find all the mutations. In this case, we have some hopes, because it seems in this work shows that there are some mutations that can be called the root mutations, mutations that most likely are generated at the very beginning of the tumor appearance. And these mutations could be good therapeutic targets to eliminate the whole tumor. But that doesn't mean that we need to find out which are these root mutations. And for that, we really need a very, very thorough molecular diagnosis. We have to characterize very well these tumors to find these targets. The other problem that we are encountering is that you can have several cells with the same mutations, but then cells can have different states of differentiation. And so you have also intratumoral heterogeneity in this sense. And the state of differentiation, what it does is changes the sensitivity of these cells to the treatment, although they have the same mutation. And this is also something that we have to take into account. Also, the presence of the stroma is very important. We have normal cells within the tumor, and the presence of those normal cells will also be very important to take into account in order to determine which is the optimal treatment for the tumor. Finally, we have the concept of cancer-initiating cells, cancerous stem cells. Within this intratumoral heterogeneity, with cells at different stages of differentiation, it seems that there are cells in some tumors, for example, in brain, breast, colon, that have characteristics that are similar to stem cells, and these cells are responsible for reinitiation of the tumor, relapse, and also metastasis. And it seems they are more resistant to standard treatments. So that means that we have also to identify those cells, characterize them, look for therapeutic target against these therapeutic targets against these cells in order to eradicate the tumor. So how are we going to do that? How are we going to analyze all these tumors? How are we going to see which are the good therapeutic targets in these different cell types, different mutations in different patients? So one way to go is to get as close as possible to the patient in order to study the tumor. And this is also based on translational research, collaboration between the clinical and basic science. And this is what we are doing in Barcelona. We are generating models that are very close to the patient. We obtain samples from the tumors, in this case in glioblastoma, We generate primary culture tumor cells, so we can have cells coming from a patient in a tissue culture, and these cells can stay there for a couple of weeks, maintaining the characteristics of the tumor cells in the patient. And then we can use these cells to treat them with different compounds to see which is the compound that works better for that particular patient. Also, we can obtain cells from these tumors and inoculate them in mice to generate tumors that we can monitor. And these tumors have the same characteristics as the tumor in the patient. The same histology, as you can see here, but most importantly, the same genomics. So if the patient tumor has mutations in p53, the same thing will happen in the patient, in the tumor. So with these animal models, we can also begin to understand how we are going to find the best treatment for a particular patient, and how we're going to get these biomarkers of response in order to find out better treatments. We can use these models as a preclinical model, and we can get several mice inoculated with the same patient tumor cells, and then treat them all with the compound that we want to analyze and see whether the compound works or not. And depending whether it works or not, that information can be translated into the patient in order to be treated with that particular compound. So that's something that is very important in order to find out which is the best tailored treatment for each patient. So in some way, we can begin to think that maybe what we should do is to take the sample of a patient, generate models, and then identify which is the optimal treatment based on the information that we get with these models. And that information can be go back to the patient and try to select the treatment. Now, as you can see, this is really costly. You cannot do this for each patient. And although you can do this and try to get a list of biomarkers, still, this is something that is going to be very tough to do. This is what we are trying to do in Barcelona, but we are using just very, very specific patients that we have that can be entering into particular clinical trials that we have, such as, for example, we are selecting based on these patients that could enter into a clinical trial with PI3K inhibitors, looking for PI3K mutations, or looking whether their cells are sensitive to PI3K inhibitors. But that is very difficult to extrapolate to many more clinical trials and patients. So this is also something that we have to keep in mind if we are thinking about the economy of all of this. So based on all what I said, I think the way to go, theoretically, is combinatory treatments. I mean, if we know now that tumor cells are able to escape to a treatment using the Darwinian selection, one will think that you need to combine treatments targeting the same target in order to lower the probability of resistance. But also, if we are thinking about the intratumoral heterogeneity, either at the level of genomics or at the level of differentiation states, which is epigenomics, we will have also to use combinatory treatments to get rid of all the cells. We will be targeted therapy, not to the patient, but to the cell, at different states of the cell, of differentiation, and also on the different subclones that the tumor might have. Now, if we think about everything together, we will need a very, very thorough molecular diagnosis at many different levels: genes, expression, protein, maybe markers in the blood. And also we will need an arsenal, a really huge number of drugs, effective drugs, that could be used in combination to treat a very small population of patients. And this obviously has a very important economical implications. And now, here is the discussion. Can we afford it, and how are we going to pay it? And I hope the next speakers will talk about this.